Now that we've excited the muscle cell, we need some way to hook that signal up to the actual contraction of the muscle, and that's excitation-contraction coupling. So there's a bunch of little steps that need to happen. We're going to stimulate the sarcolemma of the muscle cell with acetylcholine on the end plate. And then that motor end plate, if we change it enough, that electricity will then trigger the opening of electrically gated sodium channels that are just next to our motor end plate. So here's our motor end plate down here, right underneath the axon terminal. And as we use the acetylcholine and open up those ligand-gated cation channels, if we can change that electrical potential on the membrane enough, what we call our end plate potential, then the little channels next to that area, just adjacent to it, are going to open up electrically gated sodium channels. As the sodium channels open, sodium is then going to flood into the sarcolemma, and the sarcolemma will then become more positive, initiating the opening of another adjacent electrically gated sodium channel. So sodium will rush into this one, and as the membrane potential here changes, then we're going to open the electrically gated sodium channel here, and so on, just like little dominoes falling in a line. And so that wave of depolarization is going to then spread across the sarcolemma in what we call an action potential. And it's kind of painfully slow as I talk about it, but this whole process happens around 200 miles an hour on the sarcolemma, which is pretty quick. And this wave of depolarization is followed by a wave of repolarization. Remember, as those sodium channels then close, the potassium channels open, and potassium starts to flood out of the cell, making the inside of the cell more negative, and now we're repolarizing. So that action potential is going to go down or across the sarcolemma and eventually go down those little invaginations that we've talked about previously, the T-tubules or the transverse tubules. And on the transverse tubule, they're going to open up electrically gated calcium channels. Calcium channels, in this case, are embedded within the terminal cisternae of the sarcoplasmic reticulum. So as the calcium channels open, calcium is then going to flood into the center of the sarcoplasm. So to recap this a little bit, our end plate potential is altered with a ligand, which is acetylcholine. So as acetylcholine diffuses across the synapse from our synaptic terminal, we're going to open up those ligand-gated cation channels. Sodium will diffuse in, potassium diffuses out, but more sodium is coming in than potassium is leaving, and so that causes the motor end plate potential to go ahead and start to rise a bit slowly, all things considered. Now, if we don't reach enough of an electrical change to open up those adjacent voltage-gated channels, then we don't get an action potential and nothing happens. We stimulated the muscle, but not enough to actually do anything. If we do reach that threshold, and that's what it's called, is threshold, then we're going to initiate that action potential, that cascading domino effect of sequentially opening sodium channels and causing the depolarization of the membrane to zip along at 200 miles an hour. And then remember, this is followed immediately by a wave of repolarization as the sodium channels in a given area close, the potassium channels open, potassium floods out, making the inside of the muscle more negative. That cell, as it becomes more negative, is repolarized. It goes down to about -90 again. And this is the first step of repolarization. And remember, while the cell is in the middle of depolarizing, you cannot depolarize again. And so that's called the refractory period. So if we're looking at just one very specific little tiny part of the sarcolemma, we're hanging out at resting membrane potential -90, and then right here is when we have the initiation of the stimulation or the excitement of the muscle. And then we're going to have our depolarization this way, and then our repolarization. And you notice that it takes about 4-ish milliseconds or so to come all the way back to where we were. Now remember, repolarization is not only potassium flooding out of the cell and reestablishing our membrane potential, but we also need to get our ions sorted. All the sodium that's inside of the cell needs to be pumped out, while the potassium that's outside needs to be pumped in. And luckily, we've got the sodium-potassium pump to do just this. So our action potential goes down our T-tubules and opens up those electrically gated calcium channels in the terminal cisternae of the sarcoplasmic reticulum, and that releases calcium ions into the center of the cytoplasm. As the calcium is released, it is going to bind to a small regulatory protein that we find on actin, and that is called troponin. As it binds to troponin, troponin then is going to change shape slightly, and as it does so, the other regulatory protein that is connected to it, our tropomyosin, gets rotated out of the way of the actin binding sites. These are like little olive pits on our actin subunits, and as those become uncovered, then the myosin heads can come up and actually chemically engage with the actin, and that's called cross-bridging. So this whole process here is called the cross-bridge cycle. So in our first little box here, we're going to have the calcium ions go ahead and attach to troponin, which then moves the tropomyosin out of the way, revealing the actin binding sites, and then myosin will come up and cross-bridge. When it does so, we're going to detach adenosine diphosphate along with an inorganic phosphorus, and that detachment allows the cross-bridge to actually move just a little bit. The myosin heads here, you can see, are bent, whereas in the previous box, they're not. So that is called the power stroke. And that's when we actually have the attached myosin and actin powerfully move past each other and we finally contracted the muscle. Then ATP will come in, detach the myosin head, and cock it back and have it ready to go again. And so as we just cycle back and forth, this is called the cross-bridge cycle. So we have cross-bridge formation, the power stroke, the release of the myosin head, and then resetting it back to the cocked position. Remember, ATP is required to detach the myosin head and cock it back. As long as we've got high levels of intracellular calcium and ATP, we're going to continue to cross-bridge. And this is asynchronous. We're not going to have all the myosin heads attach, power stroke, and let go all in or at the same time. This happens little bits at a time. So that way we don't lose any progress we've made in sliding the actin past the myosin. And the process here is going to go ahead and shorten the sarcomere. So as our actin and myosin ratchet past each other and start to overlap a lot more, you'll notice that the sarcomere has indeed shortened. And it's not very much. You can tell that's a microscopic picture there. But when you multiply by that by several 100,000 sarcomeres in a myofibril, then you get some appreciable shortening in a muscle. So there are some issues with this. Tetanus is a spastic paralysis caused by a toxin made from Clostridium tetani, and it blocks inhibitory signals from the spinal cord, and when muscles are stimulated, they cannot stop stimulating, and that causes the whole body to kind of go into a massive spasm that never lets up. It's like dying from a whole body Charlie horse that continues on for hours until you die. And then botulism is also caused by another bacterium, the Clostridium family, Clostridium botulinum. And botulina toxin prevents the release of acetylcholine at the synaptic knobs. So in other words, a signal will go down the neuron, but then there's no release of acetylcholine, and so there's no opening of the ligand-gated cation channels, so there's no end plate depolarization, so there's no action potential, there's no release of calcium, there's no cross-bridge cycle. And so in the cases of botulina toxin or poisoning, you get the opposite effect. Instead of a spasticity, you get a flasticity, where the muscle doesn't contract at all. We can dilute this massively, and then use it for cosmetic purposes. As it's injected into muscles of the face, it causes them to become paralyzed. They can no longer contract, and so they relax, and that smooths out wrinkles. So as we terminate the signal to the sarcolemma and stop releasing acetylcholine, the acetylcholine in the synapse can either diffuse out a little bit. Most of it's going to be cleaned up by acetylcholine esterase, and that's going to go ahead and close those ligand-gated cation channels and stop our end plate depolarization. So that shuts down the action potential. That's going to close our electrically gated calcium channels on the sarcoplasmic reticulum, and then calcium can again be sequestered via calcium pumps into the terminal cisternae of the sarcoplasmic reticulum. Just to be clear, just like the sodium-potassium pump, these calcium pumps on the sarcoplasmic reticulum never ever stop. It's just that so many channels become open during a depolarization event that there's no way they can keep up. But as soon as you close those gates, they can start pumping that calcium back in and having it stay. Troponin, because calcium is no longer attached, will resume its original shape and rotate tropomyosin back over the binding sites so that myosin and actin cannot engage, and the fibers can slide passively past each other.